Hello, and today I'm going to be telling you guys what mitosis is. Now, you guys might be wondering, what are you talking about? What is mitosis? Mitosis is a process in which a cell divides into two genetically identical daughter cells. Now, you guys might be asking, why does a cell need to divide? Well, cells don't live long in our body. Some cells die after a couple days, some last a couple years. And basically, cells keep dividing. That is how cells keep going, because if all the cells in our body didn't divide, then we would die faster, just because the cells in our body keep us going. Mitosis has 4 phases: prophase, metaphase, anaphase, and telophase. Now, before we even get to mitosis, we have to talk about interphase. Now, interphase basically is a preparation for the cell. Think of it as when you're making a pizza. You have to get all the ingredients, you have to buy the dough or whatever. You have to prepare for what you're about to do. The cell prepares for mitosis 90% of its lifespan. Basically, that means it's preparing, it's bulking up, it's getting nutrients, as well as it's duplicating its DNA in order to form mitosis. Now, you guys might be wondering, where did mitosis come from? Who discovered all these different phases and all that stuff? Mitosis was discovered in frog, rabbit, and cat cornea in 1873. So let's move on to the first phase of mitosis, which is prophase. In prophase, the duplicated DNA chromatin form condenses to form chromosomes. Well, a chromosome is made up of sister chromatin. As well as in prophase, the nucleus envelope starts to break down and the mitotic spindle starts to form. And at the very top of the mitotic spindle, and think of mitotic spindles as webs, at the very top of the mitotic spindle are centrioles, and basically centrioles are what form mitotic spindles. Without the mitotic spindle, we would not be able to do this next phase, which we call metaphase. Now, in metaphase, the mitotic spindle is fully formed, and what happens is it starts to bring the chromosomes and line them up in a straight line, which we call the metaphase plate. And the reason it does this is because for our next phase, it needs to divide them equally, so that's why it lines them up in a straight line. The next phase is anaphase. Anaphase is when the chromosomes divide they basically split in two. That is why we need to duplicate our DNA in interphase, so each cell has the same amount of chromosomes. So when we're dividing this cell here, now 2 sister chromatids go to one and 2 sister chromatids go to the other. Each chromatid is now its own single-stranded chromosome. Now basically we move to our last phase, which is telophase. And in telophase, the single-stranded chromosomes unwind back into chromatin, and the mitotic spindle starts to break down and disappear because it's not needed anymore, as well as the nucleus begins to reform and it is ready for dividing. And you guys might be asking, why is it ready for dividing? Didn't it already divide into 2? And that is what we like to call cytokenis. Basically, cytokenis is what divides the cell into 2, and the reason we don't consider cytokenis one of the phases is because it happens during telophase. So basically what cytokenis does is when it pinches a cell, it creates a cleavage furrow, which contains a contractile ring. In a plant cell though, you cannot create a cleavage furrow because the plant cell is too rigid. Therefore, it creates a cell plate, and that actually divides the cell into separating the one cell into 2 genetically identical daughter cells. That is it for mitosis. I hope you guys all enjoyed this little presentation of mine. Thank you all for watching, and I hope you have a blessed day. Bye.